I'm going to talk a little bit about something I don't really work on, which is experimental work looking at the actual mechanisms involved in ADA2 deficiency, and particularly the function of ADA2 in vivo. That's the title of a paper that I wrote with my colleague Terry Tarrant, who's here, a couple of years ago. And it was based, the reason we got involved was the situation as it existed then was a little bit confusing for us, and I think maybe for many of you who are working on the disease, in that it really seems as though there are too many mechanisms that have been proposed to explain the phenotypes associated with DATA2 deficiency. And we attempted to go through the literature as it existed then and tried to look at each of these, the papers that proposed these, and try to come up with some thoughts. Let me say that our work is primarily over the years been on ADA1 deficiency, and so what I'm going to tell you now, as in the past, is colored by what we know about ADA1 deficiency and try to make some comparisons with ADA2. So we know that ADA, whether it's ADA1 or ADA2, there are two substrates and two products. Adenosine and deoxyadenosine are the substrates. Inosine and deoxyadenosine are the products. And I think when ADA2 deficiency was first discovered, and what has made an impact on the initial studies related to the function of ADA2 in vivo and pathogenesis, has to do with the idea that ADA2 is cytoplasmic, ADA1 is cytoplasmic, ADA2 is secreted. There are ADA substrates in cells and ADA substrates extracellularly, and that secreted ADA2 acts on these, and intracellular ADA1 acts on these. So the theories that we will look at have to do with ideas that existed initially about ADA2 being a growth factor, that secreted ADA2 controls the concentration of extracellular adenosine, that adenosine is pathogenic in DATA2, that secreted ADA2 derived from endothelium acts on extracellular deoxyadenosine, and the product deoxyadenosine produced intracellularly by ADA1 is pathogenic, and I'll have to get back to that. And finally, an interesting paper, a couple of papers suggesting that secreted ADA2, which, as you know, is elevated in a number of infectious and some inflammatory diseases, might actually be pathogenic in non-alcoholic fatty liver disease, which has a new name now. And that inosine, the product produced by ADA2 acting on adenosine, is pro-inflammatory. I am not going to be talking about intracellular ADA2. You're going to hear a lot about that. But I will just say that I think my view is that we're a little bit at a crossroads. I'm going to talk about secreted ADA2, but the interesting talks you're going to hear coming up are going to be about intracellular ADA2. So I showed you a diagram that I thought was a little bit simplistic, and this just shows why. All cells contain or possess an equilibrative nucleoside transporter, ENT, that can equilibrate extracellular and intracellular adenosine and deoxyadenosine and other nucleosides, so that the barrier between what's outside and inside really doesn't exist, or not as clearly as one would like to think. In addition, ADA2 is extracellular and ADA1 is intracellular, except that in humans, there is a glycoprotein that is on the membrane and secreted, or gets or released into plasma called CD26, which binds ADA1 in a very specific way. It was originally called the adenosine ADA complexing protein. It exists on cells. All of the ADA1 in plasma, and there is some, is bound to CD26, and there are many organs on which that so-called ecto-AD1 exists. So let's go into the hypotheses. The first is that ADA2 is a growth factor, and that's based on the homology between the gene for ADA1 and a gene originally discovered in flying insects that suggested that ADA2, or the homologue of ADA2, was a growth factor. And I'm not going to go into this. I've talked about it before, but basically it was called adenosine deaminase growth factor because it stimulated the growth of cultured adult insect cells in medium. It turns out that ADA-GF is really like ADA1, not ADA2. It's got a very high affinity for adenosine, unlike ADA2, human ADA2. It's not a growth factor. It really just gets rid of toxic levels of adenosine present in conditioned medium, not conditioned medium, in culture medium that's been prepared with yeast extract, which contains a lot of purines. You can substitute bovine ADA1 for ADA-GF, and it works just fine. And ADA-GF does not stimulate growth in adenosine-free minimal medium. So the original ADA-GF is not a GF. It's an ADA that looks a whole lot like ADA1. Nevertheless, there have been some very interesting studies showing that human ADA2 interacts with myeloid and lymphoid cells and has effects on their growth and maturation. These effects sometimes require active recombinant ADA2, sometimes inactive ADA2 will work, and it's not clear if this has physiologic relevance, but it's still out there and still a possibility. The second hypothesis I want to go into has to do with the idea that secreted ADA2 controls the level of extracellular adenosine. It's called an adenosine deaminase. There is extracellular adenosine. It's secreted. Why not? The problem is that the Km for ADA2 is extraordinarily high. It's around 2.5 millimolar instead of about 25 micromolar for ADA1. That extracellular adenosine concentration is submicromolar in humans. There is a mass spec method for measuring adenosine and deoxyadenosine in Guthrie cards of newborns that is widely used now to diagnose, or rather to pick up children who might be ADA1 deficient. And in studies of 50,000 newborns, they came up with a concentration of adenosine of about 0.5 micromolar. Measurements of ADA2, or adenosine levels in patients with ADA2 are very few, but using the same mass spec method on Guthrie cards, the concentration in 2 kids with ADA2 from Belgium was normal. And the NIH has looked at 9 patients, and again, I won't go into the methodology, it's different, and they found an average of 0.4 micromolar. Those are normal levels. In contrast, in kids with ADA1 deficiency, values from 4 to 25 micromolar or greater have been found, and those individuals have normal ADA2 levels. So I would just say that rigorous systematic quantitation of extracellular adenosine has not been done in ADA2 deficiency, but it would be very useful to do. So if ADA2 isn't controlling extracellular adenosine, what is? And what has to be understood is the literature talks about ADA2 extracellular and ADA1 intracellular as binary choices for what controls extracellular ADA2. But extracellular ADA2 is really a lot in blood. And blood has more than plasma. It has whole blood, is red cells. And if you quantitate the amount of ADA1 activity in red cells, it's huge compared to plasma ADA2 activity, and that's measured at 10 millimolar adenosine, saturating adenosine. And if you go down to the physiologic concentrations of adenosine, the activity of ADA2 would be minimal. So this is really a huge contribution to ADA activity in blood. And red cells have a transporter. They also have adenosine kinase as well as ADA1. And they have very high affinity for adenosine. In addition, ecto-ADA1 bound to CD26, as I said, is present in plasma and on the absorptive surfaces of many organs. And I'll just mention that mice have cytoplasmic ADA1 and a transporter, but they don't have ADA2, and in mice, CD26 does not bind ADA1, mouse ADA1, so there is no ecto-ADA1. So really, in mice, it's ADA1. Whether it's all intracellular or extracellular is another question. So, very interesting paper that you'll hear about today again is about the role of endothelial-derived ADA2 and deoxyadenosine in pathogenesis, and the postulate that postulates that ADA2 is secreted from endothelium and acts on extracellular deoxyadenosine, the other substrate for ADA. That in data 2, when ADA2 is missing, extracellular deoxyadenosine is not being deaminated extracellularly, but it can enter endothelial cells via the transporter and converted by ADA1 inside the endothelium to deoxyadenosine. And that intracellular deoxyadenosine therefore accumulates to higher levels, and through mechanisms that I think are not well defined, maybe better defined than I'm saying, inhibits the synthesis of S-adenosyl methionine, which is the universal methyl donor in cells, blocking epigenetic and genomic methylation, which results in triggering type 1 interferon response. Oops. So what about that hypothesis? So it, to me, seems implausible and unnecessary. First place, the level of adenosine is low in plasma, as I just said, but the level of deoxyadenosine in human plasma is undetectable. It's vanishingly small. In the same 50,000 Guthrie cards that were analyzed, all they could say is that it was below the limit of detectability, which was below 0.07 nanomolar. It's very low. ADA2 has a very high Km for its substrates, so it would be difficult to imagine it doing anything with deoxyadenosine, even if you could measure it. The second thing that I'll point out, and this has been known since ADA2 was discovered by Rochelle Hirschhorn and others in the 1970s and 80s, that ADA2 is even less active with deoxyadenosine than with adenosine, whereas ADA1 is equally active. And we just did a quick experiment with blood plasma from an individual who had threefold elevation of circulating ADA2 activity the other day. And we looked at the activity with 10 millimolar adenosine as a substrate and 10 millimolar deoxyadenosine as a substrate. And there's much less activity with deoxyadenosine, whereas the ADA1 in that plasma sample is equal with deoxyadenosine. Just to illustrate, that's the only data I'm going to show. So what else can we say? Is it necessary to postulate that ADA2 acts on extracellular deoxyadenosine? And I don't think so, because deamination of deoxyadenosine, wherever it is, by ADA1 is physiologic, and it's essential for lymphocyte viability and function, which you learn from the consequences of ADA1 deficiency. In addition, cells all contain a purine nucleoside phosphorylase that would eliminate deoxyadenosine, so it can't accumulate in cells. Whether ADA2 is expressed significantly by endothelial cells is controversial, and you'll see a couple of talks about that and posters. And it's been reported, at least by the group at NIH, that endothelial cells do not express ADA2, but maybe not. also, there's a very interesting paper that you might want to look at that says that both ADA1 and CD26, the complexing protein, are expressed by endothelial cells, human endothelial cells, that they're co-induced by hypoxia, and that they increase the presence of ecto-ADA1 on the surface of endothelial cells, and that's been postulated to control the level of extracellular adenosine due to inflammation to prevent chronic elevation of adenosine. So the last hypothesis I'll go through is the interesting idea that elevated levels of ADA2 are inflammatory in the setting of non-alcoholic fatty liver disease. And what they point out is that if you grade the level of fibrosis in liver biopsies of patients with non-alcoholic fatty liver disease, higher levels of fibrosis are associated both with a twofold elevation in serum ADA2 activity and with the presence of ADA2 expressing macrophages in portal regions of the liver. In experiments, they showed that exogenous ADA2 upregulated pro-inflammatory cytokines in cultured blood monocyte-derived macrophages, and that when they looked at what might be responsible for the upregulating of pro-inflammatory cytokines, they found that inosine, exogenous inosine, but not adenosine, upregulated PDGF-beta in these monocyte-derived macrophages. And they concluded that ADA2 secreted by infiltrating monocytes in the liver convert hepatic extracellular adenosine to inosine, that increased levels of inosine are a key pro-inflammatory and pro-fibrotic mediator in that disorder. All I can say about that is that it seems paradoxical that inflammation could result both from the deficiency of ADA2 in DATA2 and from elevated circulating ADA2. If that were the case, you'd have to be very careful when you treat with PEG-ADA2, maybe. Postulating that secreted ADA2 alone controls extrahepatic adenosine or inosine ignores potential effects of red cells and also hepatic ecto-ADA1. And in that context, CD826, the ADA complexing protein, is known to be expressed at high levels on hepatocytes, and hepatic CD26 levels correlate with the degree of fibrosis in fatty liver disease. So that you'd have to consider that ADA2 alone can't necessarily be responsible for producing inosine in the liver. So in summary, I don't think there's a consensus about how secreted ADA2 works in vivo. The pro-inflammatory effects have been attributed to elevated levels of ADA2 substrates, both of them, adenosine and deoxyadenosine, and to both ADA2 products, inosine and deoxyadenosine, and to both deficiency of ADA2 and elevated levels of secreted ADA2. And I think not all of those are mutually compatible. And so I think, as I said before, I think we're a little bit at a crossroads, and maybe we should start thinking about intracellular ADA2, and you'll hear a lot about that. So thank you very much.